Welcome to my channel XY Biology Academy. Now that we have understood the phase contrast microscope, let's move to one of its advanced versions, the Differential Interference Contrast microscope, commonly known as the DIC Microscope or Nomarski microscope. As we discussed in the previous topic, the phase contrast microscope allows us to observe living, unstained cells by converting phase differences into brightness differences. However, despite this major advantage, it still has some limitations. One of its main limitations is the formation of halo artifacts. Halo artifacts are bright glowing rings that appear around the edges of the specimen. These rings are not actually present in the specimen, they are optical artifacts produced by the microscope itself. Because of these halos, the boundaries of cells become less distinct, and many fine intracellular structures are difficult to observe clearly. Another limitation is that the image produced by the phase contrast microscope appears relatively flat and lacks depth. As a result, the surface details of transparent specimens cannot be appreciated properly. To overcome these limitations, the Differential Interference Contrast microscope was developed. It produces much sharper images, provides excellent contrast, greatly reduces halo artifacts, and creates an embossed or shadow-like appearance that makes transparent specimens easier to study. However, it is important to remember that this is not a true three-dimensional image. It is only an optical illusion produced by light and shadow and does not represent the actual height or depth of the specimen. The principle of the Differential Interference Contrast microscope is based on interferometry. Interferometry is a technique in which two light beams are compared after traveling through slightly different paths to detect extremely small differences that cannot normally be seen. Before understanding this principle, let's first understand polarized light. Normally, light coming from the microscope lamp vibrates in all directions perpendicular to its direction of travel. This is called unpolarized light. When this light passes through a special filter called a polarizer, only the light waves vibrating in one plane are allowed to pass. The light obtained is called plane polarized light or simply polarized light. Now let's understand the working of the Differential Interference Contrast microscope by following the path of light through its ray diagram. Light from the light source first passes through the polarizer, where ordinary unpolarized light is converted into plane polarized light. This polarized light then enters the first Nomarski modified Wollaston prism, also called the beam-splitting prism. A Wollaston prism is a special crystal prism that splits a single beam of polarized light into two separate beams. Inside this prism, the polarized light is divided into two coherent light beams called the ordinary ray, or O ray, and the extraordinary ray, or E ray. Coherent light beams mean light beams that originate from the same source and maintain a constant phase relationship while traveling. These two beams are polarized at right angles to each other and are separated by a very small distance known as the shear distance. Shear distance is the small distance between the two light beams as they travel through adjacent regions of the specimen. It is usually about 0.2 micrometers. The condenser then focuses these two beams so that they pass through two adjacent points of the specimen. As the O ray and E ray travel through the specimen, they pass through slightly different regions. If these regions differ in thickness or refractive index, each beam experiences a different optical path length. Optical path length is the effective distance traveled by light inside a material. It depends on both the thickness of the specimen and its refractive index. Because of these differences, one beam is delayed slightly more than the other, producing a small phase difference between them. After emerging from the specimen, both beams pass through the objective lens and then enter the second Nomarski modified Wollaston prism, also called the beam recombining prism. This prism brings the O ray and E ray back together into a single beam. The recombined light then passes through another polarizing filter called the analyzer. An analyzer is a second polarizing filter that converts the invisible phase differences between the two light beams into visible differences in brightness. At this stage, the two light beams interfere with each other. Interference is the interaction of two light waves when they meet. Depending on how their waves combine, the final image becomes brighter or darker. If the two light waves reinforce each other, constructive interference occurs and that region appears brighter. If they cancel each other, destructive interference occurs and that region appears darker. As a result, extremely small differences in optical path length are converted into visible light and dark regions. The final image appears as black to white shades on a gray background. One side of the specimen appears bright, while the opposite side appears dark, producing an embossed or shadow-like appearance. This makes cell boundaries and fine intracellular structures much easier to distinguish. However, remember that this shadow effect only creates the illusion of depth and does not represent the true three-dimensional structure of the specimen. The differential interference contrast microscope has several advantages. It produces high-resolution images with excellent contrast, greatly reduces the halo artifacts seen in phase contrast microscopy, and is ideal for observing living, unstained transparent specimens. However, it also has some limitations. The microscope is expensive because it requires specialized optical components such as polarizers and Nomarski prisms. In addition, the apparent shadow depends on the orientation of the prisms, so some structures may appear differently if the specimen is rotated. So, compared with the phase contrast microscope, the differential interference contrast microscope overcomes the problem of halo artifacts, produces sharper images with higher contrast, and uses polarized light along with the interference of two closely spaced light beams to reveal fine cellular details that are otherwise difficult to observe. Thanks for watching. Like this video and subscribe to my channel.